The main function of antibodies is to activate the classical pathway of the complement cascade. The complement proteins, such as C1, often involve serine protease enzymes, which cleave other proteins, parts of the complement pathway, from inactive forms to produce active forms. Interestingly, some of these, such as C1, have a variety of functions other than that depicted in the classical pathway. For example, complement C1 is also involved in neutralizing viruses, in initiating phagocytosis as an opsonin, in the removal of apoptotic cells, and in the maturation of immune cells. And thus we see that this step is not irreducibly complex. C1 did not have to appear with the antibodies of the adaptive immune system. C1 can bind some antigens without antibodies, and C1q and the homologs of C1s and R are possessed in lampreys which lack antibodies. These C1 subunits are homologous to components of the lectin pathway, and thus were produced by the duplication of genes which already existed in more primitive vertebrates involved in other functions. Complement protein C3 is the most important component of the complement cascade. When it is activated, it is capable of forming an opsonin which will attract phagocytic cells. It also then can stimulate the complement protein C5 to begin the formation of the membrane attack complex, which will put a pore into the microbial membrane. The antibodies in the classical pathway will ultimately activate complement C3 through complement protein C4b and C2a. Also, the lectin pathway, which use lectin proteins which bind to the sugars of microbes, will also activate complement protein C4b and C2a, which then activate C3. There is also a simpler alternative pathway in which other molecules of complement C3, once activated, will activate the C3, which causes the membrane attack complex to form. So there are three pathways which utilize complement protein C3 to initiate the formation of the membrane attack complex. Thus, the use of complement protein C3 in the classical cascade involving antibodies is not an example of irreducible complexity. Antibodies simply utilized a complement cascade which was already present in more primitive organisms, using lectins in the lectin pathway, or even the more simpler alternative pathway in which the complement protein C3 is capable of binding microbial antigens without either lectins or antibodies. When certain complement proteins, which are serine proteases, cleave other complement proteins, fragments are produced, and some of these fragments have functions in their own right. So fragments C4a, C3a, and C5a are called anaphylatoxins, and they will promote the release of histamine from mast cells, the dilation of arterioles, and the chemotaxis of phagocytic cells, such as neutrophils, dendritic cells, and macrophages. Note that there is no irreducible complexity here. Irreducible complexity argues that there would be no adaptive function if an organism possessed some but not all of the components of a complex system. Notice that these fragments possess function outside the complement cascade, and none of them are absolutely essential. There are other chemicals which promote vasodilation or chemotaxis or histamine release. And so therefore there can be the gradual evolution of a more complex system because more primitive organisms, which have some but not all of the components, would still have an adaptive value. The terminal members of the complement cascade, C5 through C9, are known in jawed vertebrates. They seem to have evolved from gene duplications, and these proteins belong to the same gene family, modified duplicates of an ancestral protein.